So yes, I've been using dynamic causal modeling to model organelles in midbrain neurons. So a very brief introduction to this is Parkinson's disease is driven by a buildup of a protein called alpha-synuclein, which impairs organelle function in the cells and will eventually lead to cell death. And this reduces the function and stresses the cells and will eventually kill them. So the next logical question perhaps is, what actually causes alpha-syn buildup? Why does it happen? We don't kind of. In some cases, we do know, sort of. So you take this schematic of some organelles in the cells, and we can think about certain genetic mutations, which account for about 10% of Parkinson's cases. So we kind of know what organelle are going to affect it. So, for example, an SNCA mutation might lead to more aggressive alpha-syn seeding, so you get more buildup of the protein. Or a PINK1 mutation might knock out the mitochondrial function, enabling leak. So, the very short way of clearing alpha-syn is the lysosomes will clear it, and they're powered by the mitochondria. It's a very brief way to do it. So, our data was from the Parkinson's Progression Marker Initiative and some of Chris's QMAP cohort as well. And the way we get our data is you could take a skin sample from the patient, and you can harvest the stem cells from that skin biopsy and then turn them into neurons. And that's what we image. It's a very cool process. And there's this paper. This is the group we work with at the Francis Crick Institute. And this is the kind of data they produce. And it's from a machine called an Opera Phoenix. And in addition to producing these lovely images, it also spits it out as tabular data. So it gives us a reading of the sort of fluorescence of various dyes, which correlate to the organelles we're looking at. And that's the data I've been working on, is this tabular data. I haven't done any sort of nice image processing or anything, although that is possibly in the pipeline. So yeah, we got 36 individuals who are either healthy control, had sporadic Parkinson's, or had familial Parkinson's. Additionally, 2 genetic patients had their defects corrected to make isogenic lines. So much for this research. So in total, we had about 4.5 million cells across 38 lines, and they were measured across 5 weeks. So we have 5 by 38 patients. Now, one of the things about working with these cell biologists is they love to just kind of spring compounds on you unexpectedly. So they sort of went, oh yeah, some of the cells are dead, and we have this dye. We can tell if they're like really dead or really alive, but the threshold on this, we're not very clear. It might be wrong. And they also kind of went, yeah, so this dye is called cytox green. So it could be misclassifying some cells. And yeah, this dye basically works with if the cell's dead, then the dye should get into the cell, so it gets more. So it's about the membrane integrity, which again, there's an assumption there that the cell, bad membrane intensity, dead cell, good membrane intensity, alive cell. So schematic of how it was working is this. This is what they were doing. They just pick the number, either side, they get rid of it. And then this is the pipeline I came up with. Just basically taking we can kind of tell which are the most alive and which are the most dead cells. So we take them and as our alive and dead groups, and then I trained a neural network to just discriminate between them. So that data set's about 100,000 cells in it, so it's pretty good size for a deep neural network. And we get a really accurate classifier telling us if the cells are alive or dead. And then we can also use explainable AI methods to give us some insight into what factors are actually driving the cell to be alive or dead. So we also have a problem with non-neuronal cells. And this was a bit more tricky to solve because these are originally stem cells that they're turning into neurons. Not all of those cells are going to turn into neurons. And when I asked them, I was like, well, how many are we talking here? They were like 10, 20% maybe. They wouldn't really know. So yeah, this is another problem we have to solve. And I turned to deep learning again to do something similar. That model is about 87% accurate in this binary classification, if it's a neuron or not. So between those 2 classifiers, our pipeline sort of ends up looking like this. We've removed the outliers, we've the non-neuronal, classified the death. We get about 3.27 million cells at the end. So each step is removing about that much. And yeah, I think this is a much better solution than I call it the two-hand solution they were using. So whenever I spoke to them about the cells that shouldn't be there, they kind of went like this. And whenever I showed them data, they would kind of do this. That was a problem we don't have anymore. So it's quite nice. So then as sort of, we wanted to know a bit more about the data, we decided to make a classifier just for our healthy controls. And using Chris's Lambert's work, he could make a clinical classifier to tell the patients that were going to progress the fastest. So I just made a neural network, again, very simple, just a binary classifier to say, can we tell if this is a healthy cell or a familial fast progressing cell? And we can. I looked at week 4 of our experiment, and you get 44 features out of the data, out of the Phoenix machine. And yeah, it was fairly accurate. I think it got like 85% accuracy. But that's not the most interesting part. Is we could use this thing called chat, which tells you like the most influential inputs to the model. And if we look at the top 5 most influential inputs, they are all related to either the lysosomes or the mitochondria of the cell. So this kind of tells us where the dysfunction is in this SNCA mutation, although the mutation itself is not. I should mention we didn't stain for alpha-syn on this, so we don't have data on that. But the things that are dysfunctional are the lysosomes and the mitochondria, and that's probably where we should be looking. So this brings us on to DCM. I hope you could put up with me not talking about DCM for about 6.5 minutes. I sort of came up with this very simple model as a proof of principle. And we just have our lysosomes and our mitochondria, and they are meant to be clearing the alpha-syn. And if there is too much alpha-syn or they are not functioning properly, that alpha-syn is going to impair the lysosomes and mitochondria. Well, that's our hypothesis going in. So these are the equations that sort of govern this. Again, relatively simple. Alpha-syn is bad. Lysosomes and mitochondria are good because they're going to get rid of the alpha-syn when it shouldn't be there. And again, we looked at our best cells, our healthy controls, versus our worst cells, which are familial fast progressors. So first up, the sensitivity analysis. And right off the bat, we can see that the parameters that are most sensitive across the 2 models that have been, this is at the group level, are very different between the 2. So yeah, and then if we put it on the same scale, you can see on the familial, parameters are really actually not very sensitive compared to the control. So, you know, those connections are not doing very much. There's clearly some impairment. So if we move over to Bayesian model reduction and we look at our healthy controls again, it's not really getting rid of any connections. It says the best model is this fully connected one I came up with at the start. But then when we look at the familial, there's massive impairment. And I really want to draw your attention to these red lines here. So typically mitochondria is giving the energy, it's fueling the lysosomes. It's a very non-technical explanation. And that connection is gone, it's completely impaired. And also the effect of lysosomes on alpha-syn. And we assume this connection here is mitochondria fueling other proteins that will affect alpha-syn. And they are also completely gone. So there's clearly a massive disruption of how the cell is functioning. And why, and yeah, there's clearly disruption in the familial cells compared to the healthy controls. So this is kind of, yeah, the next bit is a bit of a work in process. I was hoping I would have it ready for this, but unfortunately I didn't quite get there. So there's a few other issues here. One, this is the actual histogram of the data for healthy control in blue and familial fast progressing in red. And you can quite clearly see there's a bimodal distribution to it. So to do this previous work, I Z-scored it at the start to kind of get rid of this and ignore it for a bit. This is probably actually a really interesting underlying dynamic here about what is causing these 2 or 3 peaks, which I'm hoping to use DCM to solve. So my kind of plan, so the first thing you can do is figure Gaussian mixture model to it. You kind of end up with a result like this, and you get the mixing proportion, the mean of the 2, so you can kind of tell get an idea of it, but how do we use this in a model? So if we expand the model, because that model will be too simple to explain all the dynamics and get all the information we want about the cells, we can then expand some of the compartments. So if I look at the mitochondria, we split that into like a low, mid, and high functioning mitochondria. The dye, luckily, basically, the more intense it is, the higher functioning the mitochondria is. So basically, I want to implement this switching model where we switch between low, high, and mid states. And then you can calculate the transition probabilities. And then that, I think, would be another interesting thing to look at, comparing healthy control to familial Parkinson's cells, is like these transition probabilities and how they're working, why that happens. Our kind of working theory is that as the cells die in Parkinson's or get regressed, you're going to have lower functioning mitochondria, and that's going to, we think that's causing, and that's why we're getting these peaks. Some of the other cells that have to overproduce and overwork the mitochondria, which is why we think they're diverging. And that's the theory we're going in with, and that's the theory I kind of want to test with this model. So yeah, that's kind of where I'm at with it. This would basically end up with each of those states adding this parameter into the model, just to say if it's like in a low, mid, or high state to modulate the connections. So yeah, finally, I've just got a lot of people to say thanks to. I joined the Phil a little under a year ago. And as Peter, you might have heard from Peter's very kind introduction, I hadn't worked on like SPM or brains or anything before. So I've got to say a massive thank you to all these people for like making me hopefully a competent scientist in this field. And yeah, just for their support and help. And I've got to say a massive thank you to Chris for all the help and support he's given me over about the past 12 months. So thank you all very much for listening.